It's my pleasure to introduce Professor Sarah Cobey. Sarah is an associate professor in the Department of Ecology and Evolution in the program in biophysics at the University of Chicago. She completed her PhD in ecology and evolutionary biology at the University of Michigan and her postdoctoral training at the Harvard School of Public Health, working with Mark Lipsitch. Her group uses computational modeling to investigate the coevolution of pathogens and host adaptive immunity, and this is true even in non-SARS-CoV-2 pandemic times. Major focus of their group has been on how the antibody response to influenza shapes flu epidemiology, evolution, and vaccine effectiveness, and they have been pursuing, her and her team, similar types of questions with SARS-CoV-2. She's the recipient of major awards, including NIH New Innovator Award and the James McDonald Foundation Complex Systems Scholar Award. And since it's 9 o'clock and I don't want to steal any of her time, it's my true pleasure and delight to have Sarah kick off the plenary component here with her lecture on modeling COVID-19, what went wrong and right, and what's next. Sarah, take it away.
Thank you so much, and good morning, everyone. I am really impressed by this program. I wish I had been able to participate in a program like this when I was a graduate student. First, I want to check, can you see my screen all right?
Yes.
I'm going to take that as, okay, great. All right, so I'm thrilled to be giving what I hope is an inspiring talk on the ways that models have and haven't influenced the COVID-19 response. And one of the main goals here is to be highlighting how much work there is yet to do and how much we need fresh minds like yours to be tackling these problems. So when I think back, it's not super constructive, when I think back to frustrations that I've experienced during the COVID-19 pandemic professionally, one of the things, this is not at the top of the list, but one of the things that pops out is an attitude, really a comment that I heard repeatedly in direct conversations with policymakers and politicians, and also read in the news, sometimes even heard from other scientists. And it was this comment, oh, it's only a model. You know, so it's not necessarily a serious idea that we need to contemplate further because it's only a model. Okay, so I hope this already rankles some of you. If it doesn't rankle you, this is actually probably my primary goal for the talk, is to help you realize, and this is something I think that Professor Weitz was really just saying, you know, modeling is something all of us do. It's really just a way to reason formally when we talk about modeling as a professional activity. And so by reason formally, and I'm thinking in particular about, you know, modeling of the natural world, what we're really doing is we're making our assumptions very explicit. You know, we're saying these are the only possible, you know, transitions for hosts, so these are the only things the virus can do, and it's going to happen at these rates, and it's going to be stochastic, or it's not going to be stochastic. So, you know, it really forces us to lay out all of our assumptions and beliefs about the system. It requires, if we're doing it well, that we use math and we not just rely on our intuition. As we all know, our intuition is often not very good when it comes to stochastic nonlinear dynamical systems. We tend to be, I think, innately more linear thinkers. And then finally, and again, I think that's great that this is practically in like the charter of the program, it forces us to quantify uncertainty. And this is an area where I think all modelers can improve, especially when you know, we're interfacing with policymakers, because it's really hard to do this like fully and well. All right, so one of the bottom lines here, though, is like we're already all modelers, and by being in this sort of like program, you're very well on your way to, I think, taking modeling, you know, to the next level and to being able to reason quite well. And I think, you know, one of the main takeaways here is we still need to get better at explaining what we're doing to other people. So I'm going to talk about this kind of general problem in the context of how models have been used and abused. And again, I'll be highlighting where we go next. And by we, of course, I really mean mostly you, fresh minds who can confront these questions from like a new angle. All right, so the examples that I'm going to cover, and this is not necessarily the best set, and I hope that in the Q&A you can bring up some examples that you've run into and that stick out. We're going to talk about the role of modeling in this early question that was debated in the pandemic about, why should we flatten the curve? We'll talk about a policy question: was centralized quarantine required in Wuhan to stop the initial outbreak? Did contact tracing stand a chance in Illinois? So I'll be bringing in some Illinois examples because my group spent a lot of the last year trying to assist policymakers here. Which projections or forecasts are trustworthy? How do we know when a model is trustworthy? Which policies in general have been effective in reducing the rate of transmission, and how can we use modeling to determine that? And then finally, I'll touch on how vaccination can impact viral evolution. And I want to emphasize that for almost all of these questions, I think that there's a lot of modeling still to do. All right, so I also want to emphasize that there's a lot of different kind of modeling out there. So we'll start with a simple deterministic SAR model, which is something I think you'll be covering later today when talking about flattening the curve and the intuition there. I'll touch on methods for estimating the reproductive number in addressing this question of whether centralized quarantine is required in Wuhan. For this contact tracing question, it's really only 2 slides. Basically, the modeling here is pretty simple arithmetic, but also individual or agent-based models have been used to address this kind of question. We'll talk about the difference between mechanistic SAR type and more statistical models when talking about projections and forecasts. Combine SAR models with RT estimation methods for looking at which policies have been effective. And then again, I'll just touch on how evolutionary modeling can be incorporated with epidemiological modeling, but mostly there's a ton to do here. So the philo stands for the phylogenetic modeling there. All right, so start with this first question. So think back 14 months to, I think, when the early discussion started about, should we flatten the curve? Because some people were arguing, we should just get it over with, right, and reach herd immunity, and those kinds of arguments actually persisted for a very long time. So what I'm showing here is an image from the popular press about, the benefits of flattening the curve. So again, the idea, to flatten the curve is you introduce mitigation measures that, reduce the transmission rate, and basically your epidemic is slower, and you don't have, you can see here, like as high a peak as you would have without protective measures. And so, the horizontal dashed line is emphasizing a really critical point, which is that, our healthcare systems have finite capacity, and exceeding that capacity is likely a really bad thing to do. It's probably going to be increasing the infection fatality rate and increasing, of course, morbidity, mortality from other conditions. So this is one pretty obvious reason why it's good to flatten the curve, at least somewhat. But what I hope you'll really kind of intuit by the end of the day is that flattening the curve is beneficial too, because it can reduce the total number of infections. And actually we can see this very easily from like the simplest and most central workhorse in infectious disease modeling, which is this SAR model. So I'm sure you'll go over this in more detail later today. I'm just going to touch on the main assumptions here. What we have these are basically 3 compartments that the host population can be split into. We're assuming that they can either be susceptible, infected, and infectious, that's the I, or recovered. So we are ignoring the possibility of death. We're ignoring any kind of like latent period where people are infected but not infectious. It's real basic. But the good news is that, you can work from this sort of model, and many of the intuitions and results that you derive from it hold for more elaborate models. All right, so only 2 things can happen in this system. So susceptibles can become infected, and so we're assuming here that they're doing that by contacting infecteds. So that's what the I is there. So susceptibles have to contact infecteds, and we assume that the rate of infection and rate of infection given contact and rate of contact, we just collapse it all into this transmission rate beta. And then the only other thing that can happen is we can have infecteds recovering, and we're assuming for simplicity that they do that at a constant rate that we'll call gamma. All right, so now we've written down this compartmental diagram that represents everything, that can happen in this system. And so from these compartments and the flows, we can write down the system of differential equations. So the only thing to do for the susceptible class is to shrink, and that is determined by the rate at which these susceptibles are getting infected. The infected class, through conservation of people, is going to be growing as people become infected, and then declining as people recover, and then the recovered class can only grow in this model. All right, so what I want to draw your attention to, so one of the first things we would do with a system of equations like this often is, we want to see what the dynamics look like for some of our favorite parameter values. And so to do that, we would be integrating each of these equations, and generally we do that numerically. And so when you do that, you get something very roughly like this, though this is just something I sketched out in PowerPoint. But you see this epidemic curve. This is assuming the reproductive number is greater than 1, and we're starting, with let's say all the population susceptible, except for that one infected person. Okay, so I want to draw your attention to what's happening at the top of that curve. So obviously we have the infected compartment growing, and then it's shrinking, and at the top of the curve, it's passing through dIdt equals 0. So this is a really nice place to explore, because it's telling us like a lot of the threshold phenomena in the system that we can use to think about a lot of different features. So this is just now restating the equation from before. So, dIdt equals beta Si minus gamma I. We can just rewrite that. And so we have infecteds in the system. So what we know then about the quantity in parentheses is it has to equal 0. So in other words, that inflection point is happening when the susceptible fraction is equal to gamma over beta. So people who've seen the SIR type models before might recognize that this is 1 over the reproductive number, but that's not really important here. What's important is that we've basically derived this threshold, and so if the susceptible fraction again is less than gamma over beta, and we were to just start with one infected person, and we had S below that fraction, that means that we wouldn't have an epidemic. So we've actually derived the fraction of the population that's required for the herd immunity threshold or the vaccination threshold. But what this curve is also showing is that we're going to blow right past this threshold and continue having infections if we let the typical epidemic proceed as planned. I'm putting this kind of casually, but this is something that happy to go into more formal detail on in the Q&A. So what we're seeing here is a really critical difference between the herd immunity threshold, which is again that S equals gamma over beta, and that's that dashed blue line, and the total fraction of the population that's infected in an epidemic, again assuming that we have the reproductive number greater than 1. And so this area here, I hope you can see my mouse, this area here we refer to as basically overshoot. All right, so it's basically extra people who are getting infected and, in the real world dying who don't necessarily need to. So the idea of flattening the curve effectively is saying, well, if we can reduce the reproductive number, maybe not get it below 1, but just have it a little bit above 1, we can reduce the amount of overshoot and creep a little bit more slowly, potentially, toward a herd immunity threshold. So this is some intuition that was obviously hard for a lot of non-modelers to pick up, that follows directly from again the most basic model in infectious disease epidemiology, and there were really beautiful models that were developed to show, for instance, how we could potentially have a series of successive epidemics and that sort of thing to reduce the total fraction infected, assuming that long-term mitigation measures are infeasible, but it all kind of follows from these basic threshold. All right, so I now want to turn to another hot question from early in the pandemic, which is about what kinds of non-pharmaceutical interventions are required to stop a local spread. So there was a paper that was published in the Journal of the American Medical Association that took the time series of confirmed cases from Wuhan, and from that estimated the effective reproductive numbers. This is the number of secondary cases that are being caused on average over time. I'll return to that in a little bit. So what you can see here is estimated reproductive number with the different interventions kind of lined up on top. So we can see it started, probably above 3, and it didn't really start declining substantially until there was city lockdown, traffic suspension, home quarantine. But then, the thesis of this work is that it was really centralized quarantine, which means that people were going to, these hotels and government facilities when they were infected. That was critical for stopping spread, that it wasn't enough just to be quarantining at home. Because again, that's where we see the reproductive number dropping below 1. So Mark Lipsitch and colleagues and I, we re-estimated the reproductive number over time using a slightly different method, and found that using slightly different method with what we thought were better assumptions, you actually see the reproductive number dropping below 1 in period 3, where period 3 again does not involve any sort of centralized quarantine, it's just home quarantine. But if you're interested in this, I'd like to draw your attention to the citation that I have after Lipsitch et al., Zhao et al. I actually think they did a better job of estimating the reproductive number over time. They came up with a curve that was very similar to ours, but they used slightly different statistical approach. So how we estimate the reproductive number obviously has a really big impact on our understanding of what transmission rates are doing and what might be effectively lowering them. So I want to review a couple different, questions, and really a lot of this is still like outstanding, work to be done about the accuracy of these methods and whether uncertainty is quantified appropriately. And it's, again, another thing I think you should be paying attention to here is that, there's very, very little uncertainty in like late January, early February, and I hope that strikes you as a little suspicious. All right, so the work that I'm going to be presenting has been published in PLOS Computational Biology. There have been other really fabulous papers that actually I should drop links to that have examined RT estimation methods further. They've been published since. The person leading a lot of this work was a fabulous postdoc in my lab, Katie Gostick. But this is really like quite an international collaboration. All right, so first I want to step back and think, okay, so what are alternatives, like what are the different ways we can estimate the reproductive number? So of course we could, early in the right kind of epidemic, just be measuring it directly from contact tracing. And this is something that was done for SARS a long time ago. And of course it helps that with SARS, there's less pre-symptomatic transmission and just less asymptomatic transmission. So it's much easier to reconstruct these networks. But of course, you can see how this is not going to be feasible in many, many scenarios. So another approach to estimating the reproductive number that we'll return to is inferring it from a dynamical model. But as you can see here, as you already know from seeing the SIR model, but especially as these models increase in complexity, there are a lot of assumptions that are built into them. They're very parametric. We've got a lot of parameters, a lot of states, and so that introduces the possibility for some substantial bias. Then there are a set of approaches that make fewer assumptions that infer the reproductive number from cases, deaths, other things we'll touch on, using the generation interval, so the time from one infection event to another. And I've listed the 3 major citations here, but I think this is a very active area, and there's a lot of active area of work, and there's a lot of new stuff coming out that's important. Okay, but one of the first things I want to do is draw your attention to the fact that actually when we talk about the reproductive number, we're sometimes talking about different things. So we can define an instantaneous reproductive number, and we can think of that as the average number of new secondary infections at time t caused by individuals who are still infected at time t. So it's not really anyone's reproductive number, it's kind of the reproductive number of the population at a particular period of time. So we can contrast that with the case or cohort reproductive number, which is the average total number of secondary infections caused by an individual who becomes infected at time t. So Here's some graphics that I hope help a little bit, like, distinguish between these two definitions. So at the top here, we've got the instantaneous reproductive number. And again, it's informed by past infections. So what we're looking at, the blue line, those are the people who are currently infected at time TI. And the green arrows are showing the infectiousness of people who were infected at times, before TI, who are contributing to the current number of infections like today. And that curve is supposed to represent their differential infectiousness. So people who are maybe earlier in their infections, were infected just 4 or 5 days ago, might be much more infectious than people who had been infected 2 weeks ago. Something like that. Okay, so that's the instantaneous RT. And then the case or cohort RT is informed by future infections. So people who are infected today, how many people are they going to go on to infect? All right, so I'm going to drill down into like a little more kind of mathematical detail about how we can calculate these things, and then kind of show why it matters and some of the problems that we have calculating them given real world data. So in a traditional SIR model, this is mostly useful for people who've seen it before, but this notation is really similar to what we looked at already. That instantaneous reproductive number is basically, it's the beta S times, this is the duration of infection. So that's actually 1 over the gamma we saw in the SIR model. So it's very straightforward to calculate in a traditional SIR model. And those of us who come from that direction, this is probably what we're thinking about when we think about the effective reproductive number. But statistically, again, it can be nice not to have to assume that we understand a lot about exactly how, the disease is spread. And sometimes, it's cleaner to work from the data that we have with fewer assumptions. And so this is what Ann Corey and colleagues presented in 2013, a method for estimating the instantaneous reproductive number. Basically involves looking at the number of new infections on day T, and then looking at the number of people infected S days ago, and then considering their infectiousness if they were infected S days ago. And Betancourt and Ribeiro showed that there are some simplifying assumptions that you can make that allow you to estimate this instantaneous reproductive number very easily, but you have to assume this exponential generation interval. And we know that generation intervals, again, that's the time from one infection onset to the next, are often not exponentially distributed. So there are often costs, as we know, to making simplifying assumptions. And sometimes it's worth it. All right, so the case reproductive number, formally, it's the instantaneous reproductive number and the convolution then of that infectiousness. So again, we're looking at the average number of total secondary infections caused by an individual who becomes infected at time T. Now, practically speaking, we can calculate it, and this is a method introduced by Willinga and Tunis in 2004. We can calculate it by estimating the relative likelihood or the probabilities that case J and not some other case infected case I. So that's what we're looking at there in the bottom left. And then we can sum those probabilities to come up with the reproductive number for case J. So they're, in some ways much more elegant approaches than fitting the SIR type models, but they're still really, really hard to implement properly. So here's a demonstration. This is based on synthetic data of the difference between the case reproductive number and the instantaneous reproductive number. The case reproductive number is the dashed line. We can see that it's preceding the instantaneous reproductive number, again, because the case reproductive number is kind of looking ahead. So again, this might seem like a lot of mathematical semantics, but I hope you can also appreciate that if RT is changing rapidly and one group is using, the method of Willinga and Tunis and, calculating the case reproductive number, and another group is estimating the instantaneous reproductive number, this could look like, scientists just can't agree, And, they're telling you also fundamentally different things about the impacts of interventions. And so this was one of the issues that was raised in response to that estimation of the reproductive number required, or the policies that were required for the reproductive number to drop below 1 in Wuhan, was to be looking at the instantaneous of the case reproductive number. Okay, So I'd like to just highlight a couple challenges with RT estimation. I'm not going to go over most of this, but again, even when we don't assume that we understand a lot about the epidemiology, there are a lot of things that can go wrong that might have some policy implications. So I already mentioned that the generation interval can be misspecified, so it can change over time, but then it's important to get the mean and the variance right. We have problems with right truncation of the data. So that's just the fact that the people who are getting infected today or people who were even infected yesterday are almost certainly not observed yet. We have underreporting, which almost none of the methods that were estimating RT, at least to my knowledge over the past year, have really properly accounted for. There are also lags just built into the biology of this process and reporting delays. And I'm going to highlight those in just a bit. And of course, we also have biased data streams, and there's like no end to that discussion. Okay, so I'm gonna just touch on how lags and reporting delays can once again like make RT estimation a much more challenging procedure than it might look just from glancing at the math. All right, so obviously what we're really trying to do is we're trying to estimate like the number of infections that are happening like on any given day, but we're like almost never seeing infections as they're actually occurring. I should say we're not seeing the moment of infection. Instead, we're tending to see what's happening downstream. So we're often waiting for people to develop symptoms to then present for outpatient testing or maybe to show up at the hospital in some areas or get admitted to the ICU. And certainly early in the pandemic, one of the main things that we had to look at were deaths. And on top of that, there's often a very large delay between when the test was, actually taken and then when the data on the test result are made available to the patient or in this case, really to public health officials and modelers. So there are a lot of lags to work with. So why does this matter? It matters because it's really like blurring the epidemic dynamics, which can again distort your inference of what different things are doing to the transmission rate. So what we're looking at here are synthetic data from, again, just like really simple SIR model. And so the actual times of infection, that's the gold curves. But then we have observations of cases and deaths, and those are the green curves. And so you can see, unsurprisingly, cases and deaths follow infections. But you can see too that we have not just like an increasing delay, of course, like the more downstream we're looking, deaths compared to cases, but we're seeing the peak effectively get like smushed a little bit. And that's happening again because this is a stochastic process. So these like events are getting, effectively blurred in time. So what this means is that it's a really great idea if you can, try to, ignore as many lags as possible and just deal with like the shortest lags in the system. So that would generally be the time between infection and symptom onset, which we know to be like roughly 5 days or so for COVID. And this is something that Hong Kong was actually looking at like really beautifully. This is data just from, through like mid-July. What they're recording here are for their confirmed cases, the intervals from symptom onset to reporting. And as you can see, that improved a little bit over time. So this is really exquisite. I almost cried when I saw it because in most places, including in the United States, symptom onset is not routinely collected. Time from symptoms to testing is often not recorded either. The test turnaround time is also not recorded. And I was part of a small study that found that in 3 states, 46% of respondents said that more than 1 week passed between their seeking a test and receiving a test result. And again, many people don't even seek a test until they're symptomatic. So those are a lot of delays to deal with. So what can we do about this? What I'm showing you here and really what the purpose of that PLOS computational biology manuscript is to be working through like kind of different, what seem like often reasonable methods for, calculating RT and also like pre-processing the data and really testing, again, on synthetic data, how well they work. And I think this is a really fabulous approach to use. throughout your scientific research, which is, test whatever you're doing on synthetic data just to make sure that that you think you're doing, what you're actually doing, and also just before you go to the trouble of cleaning like real data. And, it's effectively a form of power analysis and feasibility analysis. Okay, so here we're looking at this problem of, okay, what do we do about these lags? So again, synthetic data, we've got the infections, gold curve, the observed deaths, that's the green curve. So one option is to sample from the delayed distribution and subtract. So we generally know, for instance, that there'd be about 3 weeks between the moment of infection and death, but obviously there's some, uncertainty around that and some variation around that. So you could sample from that distribution to try to reconstruct the deaths, and that's what that purple curve is. And you can see you've actually made the problem worse, and that you've actually blurred things even more because you're effectively adding more noise to the system. So what you want to do instead is something, I didn't cover the blue curve. The blue curve is what happens when you just shift the green curve to the left, and that's a better option than trying to sample from that purple curve. Another thing you can do is deconvolution. These are techniques from signal processing. I think there have been a lot of improvements on this in the context of RT estimation, but in our experience, it's very finicky as to the assumptions that you make. But it's clearly something that we need to use. All right, so I'm just going to touch very briefly on again what some of this RT exploration has meant for the ways in which we were trying to assist Illinois in its COVID-19 response. So in the United States, as we know, there were like rampant problems with testing, not just like obstacles, and remain a lot of obstacles to getting tested, but then again, there were obstacles even once you were tested to getting results in a timely way. So what that meant was that when we were estimating RT in Illinois, we had to rely most of the time on hospitals as being our early indicator, which is just a little sad. So we're looking at the mean delay from onset to hospitalization, mostly using data from other countries initially, again, because symptom onset is not collected routinely in most of the hospitals that we worked with, but we have this again from other places. We do have very good data, of course, on the delay to death in hospitals. So we were using that primarily to reconstruct the reproductive number. Eventually, we moved on to COVID-like infections, but those, of course, are a little bit, or I should say admissions, with COVID-like illness, but that's kind of difficult to work with, especially we expect that to be increasingly difficult as more respiratory viruses start complicating this sort of syndromic surveillance. But what I want to highlight too is that Jalene Jeridan, who's at Northwestern University, malaria modeling, and we worked with the state to set up a system of what we called sentinel surveillance. So what we were trying to do is look at or estimate RT from cases. So these are outpatient visits from people who have symptom onset in the last 4 days. And when I say outpatient visits, a lot of this is from community-based testing sites. We also proposed looking at the labor and delivery population. So these were, women coming in and getting swabbed. And obviously they're coming in at a rate that's not so influenced by the availability of testing or waiting times, at these testing sites. But what was interesting, in contrast to some of the papers that were published early in the pandemic, where there were clear correlations between like mobility and the prevalence in women presenting for labor and delivery, and then future case numbers, we found actually that in most of 2020, there were very poor correlate of transmission in the community, and we like to think this is because that they were taking better precautions as the pandemic proceeded. Anyway, in the interest of time, I'm not showing that data. Okay, so I want to now switch to a different topic, and I'm going to touch on this just very briefly about whether contact tracing stood a chance in Illinois. Just to give you another example, in this case of like extraordinarily simple model. So as you might recall, last spring, the major discussion about reopening from shelter in place was about how different states would implement testing, tracing, and isolating. And there were discussions about exactly how many contact tracers were needed in different areas, and this is something that Illinois was quite excited about, and they spent over $230 million setting up contact tracing. So what do we need for test, trace, and isolate to reduce transmission substantially? This is something that has been explored in many, many different models, including agent-based models, especially models that, as I'm sure you're all familiar with, we're looking at the feasibility of contact tracing apps. So we need, first of all, to have the high case ascertainment. We need to be able to catch, some fraction of infections. And I'm waving my hands a little bit here because a lot of it depends on, the other parameters, but the case ascertainment matters a lot. We need to catch those cases quickly. It's not enough to catch them when they're, in the ICU. Of course, there has to be some sort of behavioral response. It's not enough just to be tracking infections. You need people then to be isolating so they can reduce their transmission rate. You also need them to share their contacts with you, and then you need those contacts, of course, to quarantine or isolate at a good rate. And then really effective testing, tracing, isolating involves cluster busting or reverse contact tracing, where if you're working quickly enough, you can, move back to the, say, like potentially super spreading event, where somebody was infected and move forward and like that was something that was used much more heavily in Japan, I think with great success. So I'm not going to work through all of the rates here for Illinois. I'm really just going to focus on the first one, which is high case ascertainment. So testing, tracing, isolating, again, was really, in addition to mask wearing and like social distancing, was supposed to be the predominant mechanism by which the reproductive number fell, or was managed after shelter in place. But something that the modeling groups that I worked with, especially this is a slide from Jillian Girardin. So she and I discussed this a lot with the Illinois Department of Public Health. So something that we tried to emphasize again and again was that the case ascertainment was still so low that contact tracing was unlikely to be doing much to mitigate transmission. So here we're just looking at, this is a back of the envelope calculation of the case ascertainment rate among people aged 61 to 70 years old. So as you know, COVID-19 deaths were not very well ascertained early in the pandemic. Here we're making the optimistic assumption that, in May when we were reopening, that the COVID-19 deaths were perfectly ascertained, so we weren't missing any. And actually by late May, that was probably fair. Earlier in the spring, probably like 50%, 2/3 even of deaths were being missed. But let's just assume that we're catching all of the deaths. So then we can use infection fatality rates, estimated again from other populations, to calculate the likely number of people who were getting infected roughly 3 weeks, before they showed up as deaths. And then you can compare that, of course, to the number of people who are being recorded as, confirmed cases. And so using these kinds of, back of the envelope calculations, we were estimating that maybe like 10% of all SARS-CoV-2 infections were being identified. And I mentioned before a study that was performed in the spring. So actually the numbers were even worse around this time about test turnaround times, which were regularly over a week at that time. In addition, we learned that there was also very low interest in sharing contacts. So, just given the fact that the case ascertainment was so low and there was basically no chance of, isolation making a difference by the time that the test results were returned, we argued that this is potentially not the greatest investment and that it would be wiser to invest in just like expanding testing capacity and improving test turnaround times and providing support for people, who were attempting to isolate. But this kind of math, I feel, was in really short supply throughout a lot of the United States, though I'm curious about others' views here. Okay, so moving on now to a more fundamental question that we all confront, which is which models, or in this case, which projections or forecasts are trustworthy? So again, thinking back a year, we saw some really interesting models being presented by the White House. And these models, I think, in spirit, were following a really, really kind of famous model in epidemiology, or I should say law and epidemiology, known as Farr's law. So this derived from the observation that smallpox deaths over time in England and Wales kind of followed this like sort of bell curve. And so the idea was, okay, maybe we could model epidemics like following this bell curve. So it's literally just, epidemics look like bell curves. Okay, this was trotted out again in the early 1980s to project AIDS cases. So Farr's law was invoked to defend this prediction that the number of AIDS cases would peak in the late 1980s and effectively vanish, by the mid 1990s. And again, the inspiration here is, well, the curves often look like they follow this pattern, so we can just, apply this pattern. So what I'm getting at here is basically a very naive, in this case, like biologically naive statistical argument that has been made about, epidemics. And this is not what AIDS actually did in the 1990s. I'm sorry, I omitted the slide that shows the AIDS curve, but it did not go away by the mid 1990s. So this was really the spirit behind the model that, again, the White House was quite excited about. So the actual math here that was used, and I should say this model evolved a lot, so it's hard to actually even refer to it as like a single model, but last spring, really, as you can see, it says right there, the cumulative death rate for each location is assumed to follow a parameterized Gaussian error function. Okay, now the thing is, when you're trying to predict, it doesn't necessarily matter, if you have like all of the biological nuances in there, right? I mean, the bottom line of prediction is just like, can you forecast accurately what's going to happen? And it's very good, of course, I think, for biologists to be putting in their favorite biological hypotheses and seeing if they actually improve the explanation or improve the description of the data substantially. I think it's really, really common for us, especially when we're starting out, but no, I still do this. It's nice to build mechanistic models that have a lot of components to them, again, that we think, represent what's really happening, and then find that actually most of them aren't necessary. And again, a really good test of your model is to see, can it do better than almost the equivalent of throwing darts? And so I would say that Farr's law or this Gaussian error function are just like, looks kind of like this most of the time, is actually truly a very useful referent. Though, in this case, it also flew, in the face of, I think, some really well-established infectious disease modeling and theory, and was absolutely not what the vast majority of modelers in the field would consider a reasonable basis for prediction. So I am mostly, I'm going to highlight actually one more issue with this model that should have, I think, made more people quite alarmed. What we're looking at here are projections of this model. And what you can see is that the uncertainty is greatest at like the shortest times, like in the future. And then there's like actually really, really high uncertainty, going several weeks out. So this is also kind of weird. I mean, there are like situations where it's maybe logical if you have no idea what people are doing now, but you're absolutely certain that like containment is going to start in a couple of weeks. That's not a fair description, of course, of the pandemic. So this is the sort of thing that I think was really hurting the field, honestly. That this kind of logic was being pushed on people who are not so familiar with modeling and who thought that this was state of the art. So for state of the art modeling, I will refer you to the COVID-19 Forecast Hub, which is run mostly by Nick Reich, and it's a really fabulous like implicitly competition between different modeling groups to forecast. Now it's like deaths, hospitalizations, at different scales. And it came from similar efforts to forecast influenza-like illness. So there's a really nice paper out on medRxiv that I encourage you to read that examine the performance of different models. And I should say these models are not all SIR type models. Some of them have SIR components. Some of them are much more agnostic. None of them really otherwise follows the approach that I showed you before. But they make varying types of assumptions. So this paper published in medRxiv compared their performance and found, and this is like a recurring theme, that the ensemble forecast, so that's kind of the ensemble model, it takes all the other models in a weighted way like into account, is outperforming, or historically outperformed, the individual models. So this is, I think, just philosophically, it's really fun to think about that. So I've highlighted the ensemble model. It's kind of hard to see in pink because I also highlighted in purple there a model from Georgia Tech, which looks, frankly, very good insofar as the ensemble model is probably really good. So this is a really active area, obviously, of research, and I think this is a fabulous place to start if you want to think about what state of the art is, and effectively like what works, and a surprising number of the models that work very well, to me, actually do not contain a lot of sort of biological and epidemiological information that I would expect them to. Okay, so I'm now going to touch really, really briefly on what policies have been effective in Illinois, and primarily I'm flagging this as an area for much future work. So the dynamical modeling that my group did, we did a lot of RT estimation, but then we also did dynamical modeling so that we could forecast the availability of ICU and hospital beds, and just kind of drill down into the dynamics by like different regions more finely. So most of this was really done by Phil Arevalo, though the effort was like kickstarted by a lot of lab alumni, and there were lots of people who were contributing to different features of the effort. So this is also a collaboration with Civis Analytics, which was contracted by the governor's office to make data available to the modelers. It was also a collaboration with the Illinois Department of Public Health. We used some within hospital data from EPIC. And this benefited from collaboration with other modeling teams led by Jillian Girardan at Northwestern, Nigel Goldenfeld, Sergei Maslov, Chick Makel, and John Ozek at Argonne. And each of the teams used different approaches. Chick Makel and Jonathan Ozek were primarily focused on modeling dynamics in Chicago, and so they were using this really massive individual-based model that is very intensely like parameterized, and has like commute patterns and everything in it, whereas Jillian, Nigel, and I were using different types of compartmental models. So what you're seeing here is effectively just an SIR model, and we have many more compartments just for bookkeeping purposes. So we allow people to become exposed and then pre-symptomatic, and then they become infected and they can just recover on their own. They can become infected and then later die outside the hospital. They can become infected and then go to the hospital. And in the hospital, of course, there are different transitions that are possible. So this is a compartmental model that we converted to a set of stochastic differential equations. So we incorporated some demographic stochasticity, in that we allowed only whole numbers of people to make transitions between these different compartments. And then we also incorporated some type of processor environmental stochasticity, in that we allowed there to be variation in the rates. So we fitted this to census hospitalizations, deaths inside, outside the hospital, whatever we could get our hands on, except for cases because we don't trust them. And what you're looking at here are estimates of the reproductive number over time using several different methods. And the background here is showing the different policies that were in place in Illinois. This is statewide at different times. So we have some trouble estimating the effective reproductive number early in the pandemic, and a lot of that has to do with uncertainty about the excess death rate, meaning the actual COVID mortality rate in different regions. And then you can see here, so we've got shelter in place is that purple periwinkle blue color. And you can see when it was lifted, and then, the nomenclature is, of course, not very accessible. There's tier 3, tier 1, and phase 4. But these are each different, shifts in policy. And the main takeaway to me is that there aren't super clear correlations, with the exception of, the onset of shelter in place, between, shifts in official policy and changes in the effective reproductive number. And so we've done a lot of analysis. I was supervising an undergraduate who did her thesis on looking at correlates of mobility and transmission rates. And at least we're not using, again, this is mostly mobility derived from cell phone data, we're not able to find really fabulous correlates of these changes in transmission. So I'm curious what other people think about this, but I think this is probably one of the largest frontiers right now in infectious disease modeling, is really understanding the behavioral component of transmission better. Coming from flu, we generally assume people didn't really care, but clearly with COVID-19, I think understanding behavior is critical. All right, so finally, I'm going to touch on, again, another wide open frontier for infectious disease modeling, which is integrating these epidemiological, ecological dynamics with evolution and vaccination. So I'm going to address this question of how vaccination could impact viral evolution. First, let's just think about what the selective pressures are in SARS-CoV-2. So we know that evolution in general is going to be selecting any mutations, insertions, deletions, recombinations that enhance transmission, i.e. that increase fitness. It's basically the same thing right now for SARS-CoV-2. So what we expect to see are general adaptations to humans, and I think I've been seeing this. So anything that improves replication in human cells, escape from any immune response, et cetera. But then over time, we expect there to be an increasing shift toward escape from adaptive immune responses, presumably because once most of the population has immunity, the greatest improvements in transmission are going to come from being able to circumvent that immunity somewhat. So we expect to see substitutions that reduce neutralizing antibody affinity, escape at T cell epitopes, et cetera. So there's been some argument that we shouldn't be vaccinating so broadly, or we should be really careful about how we vaccinate, because vaccines could accelerate adaptation, which obviously flies in the face of no, we need to vaccinate people to save lives. So scientifically, if you were to ask, can vaccines accelerate adaptation, the answer is of course yes. They are a selective pressure, and we have already seen that in recent history. So if the escape from vaccination is complete, then we expect to see a very rapid shift in gene frequencies. So, and what I'm saying is that we would expect that the escape or non-target strains will be replacing the targeted strains. And this is something that we saw with the introductions of the pneumococcal vaccines. So that's PCV, pneumococcal conjugate vaccine. 7 had 7 pneumococcal serotypes in it, 10 had 10 serotypes. You get it. Okay, so as these vaccines were rolled out, we saw that the serotypes that were targeted by the vaccines basically disappeared, and the serotypes that were not targeted rose in prevalence because they were competing with those targeted serotypes, but not in a way, or I should say, they're competing through natural infection with those targeted serotypes, but the immunity induced by the vaccine is extremely serotype specific. And so basically they experience a little bit of competitive release. So, these serotypes were pre-existing, so we're not seeing the evolution of new serotypes and that sort of thing, but it is clearly, it's evolution. Another place where vaccine-induced evolution has been observed is Marek's disease virus in chickens. So this is a herpes virus, and there's really strong evidence that vaccination increased the virulence of the virus. So, it's absolutely possible that vaccination can accelerate adaptation. So does that mean that we shouldn't vaccinate unless we can ensure there's going to be 100% efficacy or 90% efficacy? And this has been raised in particular in the context of strategies to, it's known as dose sparing, to basically spread, say, mRNA vaccine over more people, prioritizing, primary coverage. So for instance, the basic intuition here is that, if you can protect one person with 95%, effectiveness against infection, or you could take that same vaccine and split it, between two people and protect them each 60%, effectiveness, you're going to be saving more lives, and you're going to probably be, reducing the prevalence of infection and disease more if you were to split up those vaccine doses. But again, some people are saying, oh no, like this could increase instead the rate of antigenic escape. So this is a very, very strong claim, because it's basically saying, too, that it's better to sacrifice lives now to prevent the possibility that variants are going to emerge that we will not be able to handle in the future. All right, so some of the models of this dose sparing that I described, they're basically SIR type models, and they're really beautiful, and they're very clean, and they show clearly like the thresholds at which it makes sense to split a vaccine among more people. Of course, this is something that's already happening in the UK and Canada. They've decided to space for the AstraZeneca, Pfizer, like other vaccines, the interval from 3 weeks to around 4 weeks, or excuse me, that's totally wrong, to up to around 12 weeks. And of course, all COVID-19 vaccines are imperfect. So, if dose sparing is dangerous, then does that mean we should be using vaccines that have even lower effectiveness potentially, like some of the vaccines that are coming from China? So obviously we don't have perfect information to predict evolutionary impacts with confidence, but we're in a position where we have to make decisions. Good evidence exists, and this is an opportunity, again, for formal reasoning and modeling. I think that's more like really important than just conveying, intuition about how like antibiotic resistance affects evolution, or how antibiotic usage affects the evolution of resistance, and saying, oh, that's going to be completely analogous, to what vaccines might be doing. All right, all that said, I think this is still an area where there is a need for modeling, and I'm going to be outlining mostly a verbal argument that I think needs to be carried forward. All right, so when we think about how vaccine resistance could work for SARS-CoV-2, really there are two main scales to think about. The first, we have selection within hosts. So this selection refers to the generation by mutation and then amplification or spread of new variants during the course of an infection. But then also there's a lot of selection that's happening at the population level in terms of which variants or which strains are able to transmit onward, and the rate of, effectively the reproductive number, the rate at which they're able to do that. All right, so this first scenario that I've sketched out here is basically showing this example of serotype replacement. So let's pretend we just have a wild type strain. So this is like the original strain, and then we have some sort of variant that's able to escape vaccine-induced, or excuse me, able to escape vaccination. So we assume that, person infected with the wild type tries infecting 2 people, a person infected with the variant tries infecting 2 people. Let's pretend that like 2/3 of the population is vaccinated. Doesn't really matter for this example. So what we see, of course, is the wild type is not going to be able to be transmitted to the vaccinated person. So this is how the variant spreads. So this is just an example of selection happening. And again, this is strain replacement. And here the prevalence is unchanged. Now, what I think is a more realistic assumption to be making is actually that the effectiveness of the vaccine is still positive. So in other words, this vaccine escape is not complete. So what we're looking at here, the difference between these two figures is really what's happening over here, where we have, the variant that is still facing some sort of fitness cost. And again, the idea is that the immunity induced by the vaccine is still probably effective against a lot of the variants. So what we would get in this scenario is a reduction in transmission and prevalence. And here I'm going to refer to a lot of modeling in evolutionary biology and population genetics that shows that in very small populations, we know that selection is inefficient due to genetic drift. So as we reduce the prevalence, we should be reducing the efficiency with which new variants can displace one another. We know with lower prevalence, there are going to be fewer opportunities for beneficial mutations, and that's going to have knock-on effects. And there's some really beautiful modeling here by Michael Desai, Dan Fisher. Ben Good has also done some work in this area on the dynamics of what's known as like clonal interference. So we'll also we know that with lower reproductive numbers, so like lower rate of growth, beneficial mutations are less likely to spread, or really like less likely to fix. And that comes also from some modeling work done by Sally Otto and colleagues several decades ago. So there's really a lot of like really solid quantitative reasoning on what the effects of reduced prevalence and transmission should be. And as long as the effectiveness of the vaccine is still like above zero, even for the variants, there's an argument then that we might expect actually the rate of antigenic evolution to slow, at least at the population level. And if you're really interested in this, this is actually a question that my group was working on before SARS-CoV-2. We were curious whether hypothetically, if influenza vaccine were given to a lot of the world, whether we might see a reduction in prevalence. There were definitely, reduction in the rate of antigenic evolution. And there are definitely some models that argued it might accelerate antigenic evolution. So this is actually an individual-based model because we wanted to leave the evolution antigenically of these strains basically unconstrained, as opposed to saying, let's assume there's like one variant, one wild type. And what we found again and again,
Oh, sorry, I'm going to jump in just because we're at 10. So just because it's not, we do have a buffer.
2 more slides. That's okay. 3 more slides. Perfect. Okay, yeah. Perfect. Okay. Yeah, no, thank you, Josh. Okay, so what we found in this individual-based model very reliably was that vaccination could reduce the rate of antigenic evolution. All right, but then finally we have this idea, okay, well, what's going on within host? Maybe we're still increasing the rate of antigenic evolution within host so much that there are just going to be like all these new variants popping out. And this is something that had also been explored in an SIR, well, pretty complicated model. I'm not going to get into the details. They argued basically that the net adaptation rate within a host is likeliest, okay, so what we're looking at here is what happens as you increase immune pressure and how that impacts the net viral adaptation rate. So we're looking at the strength of selection, of course, is increasing as you increase immune pressure, but at the same time, the viral population is decreasing. And so this red curve is the net viral adaptation rate. And so the idea is, with 2 doses, immunity is really strong. There's going to be very little adaptation. Others have argued that possibly if you modify these dosing schedules, then we have much higher rate of adaptation. We're arguing, hence we've got this like notation here, this is unlikely, that what's more likely is that with some sort of dose sparing, actually, the net adaptation rate is still not going to be that high. And the reason is that, we see that most transmission of SARS-CoV-2, like flu, it's occurring only a few days after infection, before a strong adaptive immune response develops. We've repeatedly seen, there are actually more references than this, no evidence of selection in vaccinated individuals with influenza, and really just very little adaptive evolution in general with influenza. And we're only seeing appreciable diversity in immunocompromised patients. So this does mean that maybe, they need to be targeted. So I'm sketching out here an argument really that involves like a within host model, a between host model, not only SIR dynamics, but also the dynamics of mutation and like competing clades. So this is not a trivial model. But our hypothesis is that imperfect vaccination is probably not going to accelerate escape because this within host selection is inefficient, and the main benefit is going to be this reduced transmission at the population level. So this implies there's no reasons to withhold vaccination, and minimizing global viral prevalence could be reducing the rate at which we see new variants. They're my co-authors. Last slide. Where can we improve? So I'm mostly using this as a kickoff point for discussion. I've highlighted that there are a lot of challenges to estimating the reproductive number well, and one of those challenges actually is really just integrating data from multiple streams that are often biased in different ways. I think this is a huge one. This is really some basic science that needs to be worked out, and it's going to take, I think, some pretty advanced modeling. We need to figure out the relationship between transmission and behavior and seasonality. Finally, we also need to understand the impact of infection and vaccination on susceptibility and infectiousness. Some of this can be estimated, through like household studies and the like, but it's also going to be an area where I think we need to use some modeling and inference to make sure the patterns are matching up at an individual level and a population level. And I've also touched on this problem too of the need to incorporate evolutionary modeling into these SIR type models. And communicating model uncertainty is a perennial problem. So I'm just going to end by asking you what you think is missing and what jumps out to you, and also thanking you for your patience. And I'm sorry to eat into the Q&A. Thank you.
Thank you, Sarah, for this inspiring start and plenary. And there are actually a number of questions, and we're going to have Ethan Wold, one of the QBio students, moderate the Q&A, let you take a drink of water before we begin on the Q&A. And just one announcement, because at 10:15 we will be breaking and afterwards going into the individual Blue Jeans links that are separate from this. But Dr. Jessica Irons wanted to remind you that we will have this event link available if you have questions during the course of the next day and a half. It won't be on recording on because we're not doing a live broadcast, but if you just have a question, we'll try to monitor this as well. Okay, with that, again, let's go to the Q&A. And Ethan, if you could take it away.
Sure. Can you hear me?
Yeah.
We can hear you.
Great. Thanks, Professor Cobey, for that awesome talk. I'm going to start with, there's one question, there's a couple questions that were in the chat, and then there's also a couple questions in the Q&A. So the first question we have is, why is it important that the methods for reproductive number estimates implicitly account for underreporting if the reproductive number does properly account for the growth rate of infections? In other words, in a scenario with constant case ascertainment, is it really necessary to explicitly incorporate this into the assumptions of the estimation method?
So I welcome divergent views on this, but I don't think it's going to affect the accuracy of RT, but it certainly is going to affect the uncertainty in RT. And I think that is important to be conveying. Yeah.
Okay, great, thank you. So there was another question, what is the role of asymptomatic cases in the model, but there was no specifying what model. So maybe you could post that in the Q&A if that was your question and add a little bit more detail. So we'll move on to the...
And actually, Ethan, maybe I'll jump in because I think at that point you had put together in Zooting Q, if you want to post that in the Q&A again, Sarah, I think that had to do with your more complex model that you use.
Illinois.
Correct. Correct.
Yeah, that's a really great question. So what we were waiting for most of 2020 was cross-sectional seroprevalence data that would help us estimate the rate at which, we had asymptomatic and just infections happening outside the hospital. Never materialized. State chose not to invest in that, and we couldn't get funding for it. So basically, they don't have that much of a role. Obviously, they're contributing to the fraction that are recovered, in the model, which then of course affects RT. But we're not really using them. Really, we're actually just paying attention ultimately now to the fraction of infections that are just winding up in the hospital.
Okay, the next question is it fair to assume that underreporting is less common with deaths data than it is in case data? Presumably more people who have died from the disease were tested. Is this assumption common in the field, and how would we include it in a model?
Yeah, that assumption is extremely common in the field. I think most modelers were relying on deaths, especially early in 2020, because we knew there were no tests for a long time, and then the tests were extremely hard to come by. And even since then, once tests were more common, it's been very hard to know, and that's why these kinds of surveys that I mentioned are so useful. How many people are still deciding not to get tested because they heard there was an 8-hour limit or wait or something like that. So yes, we were fitting our model entirely to deaths for the first couple months. Eventually, we had ICU and hospital census, so those were confirmed infections, people in the hospital, and so then we fitted to those as well, and those became again the main populations that we were fitting to because the numbers are bigger. It's a slightly better reflection of what's happening in the community. So I know of extremely few groups that were really fitting to cases unless they were trying to estimate.
Okay, awesome. The next question is, do you have any insights on why the second wave in India has been so much more rapid than the first? And in general, why would a second wave cause so many more cases than a first wave?
I really wish I had a good answer to that. I don't know how much we can have a discussion here, but you know, there's some suggestion that B1617 might be more transmissible. It certainly has some of the mutations that are associated with what appear to be more transmissible variants, especially subclade 2. But no, I only know of anecdotes about behavior, and that's one of the things I find most frustrating about this pandemic is not actually having metrics of behavior, you know, to be testing out these hypotheses about what is causing changes in transmission. I also am kind of proud and sad to report I still don't understand how seasonality really works. Like, I don't understand why, you know, we have certain waves when we do. In Illinois, we had a big outbreak in October, November. Pretty much everyone, you know, was expecting things to get worse in the fall, but then it's not obvious that there were large behavioral changes. Maybe there were, but we don't have data on it, that led to the decline, and then basically like no, you know, increase in RT until like more recently. So I don't have a good answer for that. It's a good question.
Awesome, thank you. Okay, next question is, do you think a strong or weak selection pressure would be more dangerous for emergence of variants? For example, an effective vaccine that is developed or administered quickly might have a very strong selection pressure.
Yeah, I mean, ultimately the selective pressure is really, it's quite determined by the transmission rate, you know, so like ultimately like what you want to do is be reducing just the growth rates. And so I don't know if that's answering the question, but that's like the bottom line. So if there's any the vaccine that's strongly effective against something, we should be using it.
And I think, Sarah, maybe if I were to jump in,
Sure,
that question I think popped up when you showed your curve, the upside down parabola. So I think maybe part of that actually gave some of the answer there, that, you know, with respect to the two factors that are multiplying together.
Yeah,
that's probably where that question popped up.
Okay.
Great. So our next question is, contact tracing was empirically shown to be of lower efficacy with increasing disease transmission. Is there any insight from modeling that can inform about efficacy thresholds for contact tracing?
Yeah, actually, and I can't remember the citations off the top of my head, but there have been some really wonderful models, again, often looking at contact tracing in the context of phone apps, examining, you know, and often there's like, it's large parameter space, right? There are like a lot of different like delays in there. And they have worked out exactly these kinds of thresholds. And I don't want to be trashing contact tracing per se. It's been exceedingly effective, you know, like Korea and Hong Kong and many places. And in general, yes, we expect it to be much more effective as the prevalence falls. But I can't rattle off like a isocline off the top of my head. But there are papers on this, really nice ones.
Awesome. Okay, our next question is, here in Brazil, we observed 2 waves of reported infections with similar peaks, but the death curve was greater and hospitals were over capacity, probably due to a P1 variant. So how should compartmental models account for selection pressures and changes in dynamics?
Yeah, so it's tricky because there's still a lot we need to understand about P1, right? So one of the first things is you have to make models with a lot of different assumptions and test them out and try as hard as you can to find other lines of data, not just, you know, death and case curves to inform those models. So it's quite common in like SAR models, or basically there's this huge part of the field that's kind of dedicated to multi-strain SAR models, where you allow strains to be competing for susceptible hosts. And there are a lot of different ways to represent that competition that can have very large impacts actually on what you what you infer. So you could have the strains like reducing the susceptibility of the other strain in hosts that they've already infected, or reducing the infectiousness of the other strain. So there's like the mathematical machinery to do that. Once you start increasing model complexity, of course, it starts becoming really hard to to fit, to like identify like what the truth is, because you can have lots of different models that all do a great job of like reproducing the data. So one of the things that concerns me about some of the inference that's been done in Brazil so far is, again, the fact that, a lot of the seroprevalence surveys that are done, it's just the nature of like epidemiological research. Like you get this like real bias, enrollment bias effectively, and like who's being sampled. And that's even true when you're trying to then adjust, for the people you were able to measure, like using different kinds of weights. So I think there's, the more you can constrain, parameters like, the initial fraction of the population with immunity, or, the potential mechanisms of like cross immunity or competition between the strains, the easier it gets. So I think we're going to be seeing a lot of these models in the next couple of years, at least I hope we are. And I think it's actually going to be fabulous, really, for just understanding the dynamics of lots of other pathogens.
Okay, and I think that I'm going to skip around just because this question has a lot of likes and is on a different topic. At the beginning of the talk, you mentioned your frustration with policymakers, saying it's only a model. Have you found any particular strategies to be helpful in communicating the value of models specifically to policymakers, and what do you wish more people knew about models?
Well, one thing that I was a little slower to recognize than I should have been is just really like how constrained policymakers are. And I don't just mean like in terms of their time and their energy, but also the options available to them, are often like fewer, than what we imagine. So it's always easier to like communicate the importance of a modeling result, of course, if you can relate it to their interests. And so that's something that we tried to do more and more. And actually, Jalene Jarradan at Northwestern, she actually did start spending time on, the kind of economic side of a lot of these policies, showing actually it could be really, really cost effective to invest in this. And honestly, we wish we could have provided a boot camp kind of like this for a lot of the policymakers earlier in the pandemic, but like they were already too busy at that point. So there's a lot that we should be doing to increase our infrastructure.
Ethan, maybe I will jump in there and join on behalf of all the folks in the audience and just offer Sarah a round of applause. It's kind of strange to do that in these virtual formats.